The purpose of this podcast is to educate and to inform. This is not a substitute for professional medical care and is not intended for use in the diagnosis or treatment of individual conditions. Guests on this podcast express their own opinions, experience, and conclusions. The mention of any product, service, organization, activity, or therapy should not be construed as an ASCO endorsement.
Welcome to this ASCO in Action podcast. This is ASCO's monthly podcast series where we aim to explore policy and practice issues that have an impact on oncologists, the entire cancer care delivery team, and of course, most importantly, the patients we take care of, people with cancer. My name is Clifford Hudis, and I am the CEO of ASCO. I serve as the host of the ASCO in Action podcast series. For today's podcast, I am really excited to have as my guest ASCO's current president, Dr. Bruce Johnson. Dr. Johnson is the Chief Clinical Research Officer and Institute Physician at the Dana-Farber Cancer Institute. He is a professor of medicine at the Harvard Medical School, and he is probably best known to many of you as the director of the Dana-Farber Harvard Cancer Center Lung Cancer Program. Now, our conversation today is going to focus on a passion of his, precision medicine. This is, in fact, the theme that he selected for his year as ASCO president, and you'll hear more about it at our upcoming annual meeting. ASCO recently held an event focused on precision medicine at the National Press Club. This was part of the rollout of our new State of the Cancer in America event series. And Dr. Johnson was our host. We focused on extending the reach of precision medicine and addressing some of the challenges that exist in making it even more available to more people with cancer. So I'm really delighted to welcome Dr. Johnson and thank you for joining me today.
Yeah, thank you, Cliff.
So let's just begin with a level set. And we actually did this, I think, at the National Press Club as well. But what is precision medicine? How do you define it?
Well, I'm going to use the NIH definition and then expand on this a little bit. If you take a look on the website, it says that precision medicine is defined as an emerging approach for disease treatment and prevention that takes into account individual variability in genes, the environment, and lifestyle for each person. And I'm going to adopt this from one of the things you say, Cliff, and that is that it's the right drug for the right patient at the right time.
And so that probably in many ways has been our aim for decades in oncology, yet there's obviously growing excitement around the concept of precision medicine in cancer care right now. Why do you think that is?
Well, I think the things that are driving the level of interest in precision medicine and in cancer care is something that builds on the findings from the Cancer Genome Atlas. The Cancer Genome Atlas was a program that meant to define the genomic changes that are unique to different cancers as a long program that started in the 2000s. And once they ended up developing a roadmap for the genomic changes in cancer, it led to an effort to begin developing specific therapies that are targeted against those genomic changes. Therefore, once one can get a roadmap, and we've learned that things that look similar under the microscope can actually have different genomic changes that are driving the changes in the actual tumor that require different medicines for different situations.
So it's fair to say that while our goal has not changed, our tools to deliver on that goal have evolved greatly in the last few years. Is that the real bottom line, you think?
Yeah, it certainly has changed a great deal, and that's really what's driving this change in precision medicine.
And can you just describe for, obviously we have a range of practitioners and others in the audience, what are some of the concrete, tangible ways in which therapy, for example, has changed with this evolution? What are the differences in the therapeutic options that patients and practitioners need to consider?
One of the things I'm going to do is go through one disease where there's been rather dramatic changes, and the precision medicine has really changed the landscape, and that's the area where I've worked in, and that's lung cancer. I also want to give an example of some place that's a bit older that drove, and that's breast cancer. And lastly, I want to give an example in colon cancer, where we use a negative selection factor. One of the things that happened is that we gave similar therapies to lung cancer patients and had very similar outcomes for about 2 decades in the 1990s and 2000s. And then there was a discovery of a genomic change in lung cancer, the EGFR mutations, that took place in 2004, and the realization that you could treat it with inhibitors of the epidermal growth factor receptor, and they ended up working about twice as long, and we've learned that the patients who get this treatment live about 2.5 years longer. Since that initial discovery in 2004, there's been 3 additional genomic changes that we found that have been able to help predict for the efficacy of targeted agents, and that is ALK rearrangements, which are about 5% of lung cancers, and then 2 that are relatively rare. Those are the ROS1 rearrangements, present in 1%, and then BRAF mutations, a mutation commonly present in melanoma, where it's first developed, but also 1% of patients with lung cancer have a genomic change called the BRAF mutation that allows them to be treated. I think one of the ones that you've been very involved with is an observation made in the 1980s that a subset of breast cancers have multiple copies of the HER2 gene, present in about 20% of breast cancers, where one can administer agents directed against that potentially target Herceptin or trastuzumab. And last one I want to give an example is that it was discovered that an antibody directed against the epidermal growth factor receptor, the one that I talked about before that has mutations in lung cancer, is that they used it for treating colon cancer. And one of the things they learned from studying the genomic changes in colon cancer is found out that those patients who have a mutation in one of the most common oncogenes present in all cancers, that is a KRAS mutation, and those patients who have KRAS mutations do not have any benefit from getting the antibodies directed against the epidermal growth factor receptor, panitumumab and cetuximab. So that precision medicine means that you can use it as a negative selection factor, and those patients don't benefit from it, number one. Number two is you can avoid those side effects.
So I think your quick summary of those 3 common solid tumors highlights both the promise of this revolution, but also I think it can be a little bit intimidating for at least some clinicians. How do you describe the role of genomic testing in precision medicine at the clinical level? What I mean very specifically is how do physicians know when to send this testing, and then, of course, the follow-on is how best to use the results?
Well, at the current time, there's a limited number of cancers where there's approved agents for cancers, and one of the ones, the area, as I mentioned before, that I work in, there's four genetic changes for which there's FDA-approved agents, and so we recommend that the non-squamous, non-small cell lung cancers all get tested. After that, there's a number where you end up needing to do tests for specific genes. So, for instance, in melanoma, it's important to do testing for BRAF mutations where there's approved agents, as well as c-KIT mutations. So there's a couple there. The other thing that we've learned, too, is that now in colon cancer, I mentioned the example of KRAS mutations. And in addition to KRAS mutations, we now have an approval for the checkpoint inhibitors with microsatellite unstable high lesions where you can where you need to test this, and you can test it by panels. Now, one of the things that happens for our for recommending when you do this for our clinicians is that ASCO publishes guidelines to help people decide on this. There's also different guidelines that give some information for doing precision medicine, and we'll be covering this a good bit in our annual meeting that's upcoming in June.
So I think a related question, and maybe it's drilling down on this a little bit, is how does the responsible clinician and the entire clinical care team keep up with this? What do you suggest they do to make sure that they're not missing opportunities, for example, to test appropriately, but also to avoid over-testing and obviously wasting some resources? What do you recommend?
Well, one of the things we recommend is, once again, is to follow the guidelines, to go to the annual meetings. One of the things that we want people to keep in mind is that there are tumors where at the current time that are common, where there aren't any specific precision medicine therapeutic activities that one can target. So, for instance, pancreatic cancer, although it has lots of KRAS mutations, we don't have an effective agent for that. You don't particularly need to test this. At the current time for breast cancers, there aren't particular lesions that one can target other than HER2 amplification, so you can do an isolated test. Well, the way we encourage people to keep up on this is follow the guidelines. For instance, the NCCN guidelines give a lot of information. As I mentioned, our ASCO guidelines also give information on this. And also, one can also contact the academic folks who follow this and also seek second opinions of places where they not only have the capability of doing the testing, but also have a broad portfolio where people can refer people to these specific tests. The other thing I would add, too, is ASCO's own program, TAPER, that allows access to both academic as well as community oncology practices to have access to these targeted treatments.
Yeah, I'm glad you mentioned TAPER, because I think TAPER does fulfill a very specific gap and addresses a problem that I think is pretty common right now across the country, and that is the testing may be done with great intention in an area where there might not be an FDA-approved targeted agent. A lesion is identified, it is called actionable, and then a drug makes good sense to give there. And while the history of medical oncology in the U.S. included an expectation of routine off-label use, that is no longer possible because of external constraints, mostly, of course, around the area of payment. So TAPER allows us to make matches for those patients and essentially conduct a series of small histology, genomic change, targeted agent phase 2s, and we either will recognize no activity, which I think is an important contribution, or we'll recognize the potential for activity, and that provides more evidence for perhaps a larger purpose-built study or an expanded label and what have you. So I know that's a long comment from the moderator, but it's a really critical point you raise about TAPER. I want to, though, ask you for one other point in this regard, which is, what do you see as might be the value of clinical decision support? It seems to me, from a computer sciences point of view, this is a likely place for success. Have you had any experience with that yet?
Well, number one, we have our own experience at our own institution for the clinical support of this, and end up embedding this within guidelines for therapy. At the current time for us, we end up having to manually enter the genomic findings that feed into our pathway for the major diseases here at our own institution. We would like to get this to the point where within the electronic medical record, as one begins ordering the systemic treatments planned, is that the genomic testing is automatically populated, and when you go through the choices, it'll prompt you to give the agent of choice. So, for instance, one of the things that's happened is that we've gone through a number of different inhibitors of the second most common genomic driver in lung cancer, that is an ALK rearrangement, and the initially approved drug was crizotinib, but it appears that there's a newer drug where this was presented at last year's annual meeting, that a new drug called alectinib looks like it is working better than crizotinib. And one of the things that happens is that not all of our practitioners would not necessarily know this, so what we really want for the clinical decision support is that if you would end up going through that, it would know that the patient had an ALK rearrangement, and then if you tried to order crizotinib, it would prompt you to order alectinib as an example. One of the other things that ASCO is working on is the system CancerLink. which is accessible to both academic as well as community oncology practices, that ends up reading electronic records, and we hope to be able to have clinical decision support for both interpreting as well as ordering the appropriate therapy needed for the precision medicine.
Yeah, it does seem like an area where CDS, clinical decision support, ultimately should be very useful given the broad educational and operational challenges we've been talking about. One thought occurs to me that I think you could help clarify. Part of the reason that taper is needed, and I think one of the drivers of some level of head scratching on the part of clinicians is, while they may need 1 or 2 or 3 genes tested, as you describe in lung cancer, I think the clinical experience mostly is that they end up receiving the results of very long multi-panel gene tests. And so they get many, many results, I think some of which are relevant and some of which might not be. Can you just explain to everybody here why it is that they receive these long genetic or rather genomic reports when they really are just looking for 1 or 2 results?
I think one of the things that happens in our own personal experience is that when you test a solid tumor, typically these large panels will test between 2 and 500 or 600 genes. So you get a large number, and in a typical solid tumor, within those panel of genomic changes, you'll get somewhere between 5 and up to 20 genomic changes. And at any given time, when we discuss about things that are actionable, there will be trials for these. So, for instance, KRAS, which, as I mentioned before, is one of the real common ones. And there's lots of clinical trials for KRAS. However, the thing that those don't necessarily say is that although there are trials for them, thus far, it's not clear that there's any effective therapy for it. So you end up getting this report that says, they have this KRAS gene. It's actionable by referring them or putting them on this clinical trial testing agents directed against KRAS, but it doesn't give you enough information to know that there's not an effective therapy, and you probably wouldn't go to great lengths to send a patient off for a KRAS trial at someplace distant, nor would you do the effort to open the trial until there's some sort of clinical evidence that it's somewhat effective. And this is one place where the clinical decision support can be very helpful. One of the things that we think that is important for the development of guidelines is keeping that information up to date, what's actionable and what's useful, meaning that although things can be actionable, and one of the things they consider actionable is refer for a clinical trial, but what you really want to know is the agent against that particular driver, is there enough information that you could say that the person is likely to benefit from the therapeutic intervention?
Sure, and again, that's one of the things that all of these trials, MATCH, TAPER, and others are trying to resolve. But is it, I think for the average person who doesn't spend all their time in it, is it fair to say that a large number of the genes are there because essentially once you've done a few, it doesn't cost any more to do these large panels, and so clinicians should just recognize that they're not paying more, but they're getting many more results than anybody thinks they need in these multi-panel gene tests right now. Is that a fair assessment?
That's a fair assessment, and one of the things I wanted to say about this is that you get these large numbers of genes available, and we began testing systematically our patients starting in 2004 for EGFR mutations, and we started. And one of the things that is very problematic is that it's very difficult once you've done one test to go back. And if you have to do another test, if there's a new indication. So our experience was going from EGFR mutation testing in 2004 to adding ALK rearrangement testing in 2008. And to have to go back and find the tissue from the original biopsy, send it off to another diagnostic company is a real challenge. The second thing that we've learned is that the kind of magic number is that once you get to about 4 or 5 different genomic tests, the cost of doing one of these large panels is about the same. And when I say large panels, I'm talking about one that generates the results from hundreds of genes. When you factor in the cost, it's about the same to do the panel of several hundred genes as it is to do the 3 or 4 that are indicated. And that's really what drove us. The other part that one of the things that happens, and I'm using lung as an example, not only because it's where I work, but also because it's where there are the most genomic changes that can be targeted. One of the things that we anticipate is that there's 2 additional changes where we think there's likely to be FDA-approved drugs sometime in the next 1 to 3 years, and that's exon 14 skip mutations in a gene called MET, as well as NTRK rearrangements. And having those tests and having available, if there's an approved drug or if you can put patients on, it will be useful both in the academic as well as in the community practice.
So that's actually a great segue to what is our last topic, and that is challenges. And I know that you're passionate about making sure that these exciting advances are made broadly available. It's an issue of disparities and fairness and justice. But what do you think are the biggest challenges, given the advances that we've made in converting the promise of precision medicine into a reality for everybody who needs it in the U.S. and even around the world?
Well, one of the things in my theme is, it's delivering discoveries, expanding the reach of precision medicine. And both you and I have had the privilege of working in large academic centers that have had this testing available for more than a decade. And one of the things that happens, more than 80% of our patients in the U.S. Are treated outside of academic settings. And one of the things that we think is critical is, number one, is educating people about this. Number two is that giving information about how to interpret these. I think you talked a little bit about getting these reports back that would have somewhere between 10 to 20 different genomic changes, and knowing which of these are important, which are actionable now, which could be actionable in the next several years. So our goal is to work through CancerLink to make this information available at the point of care. The second is working with the electronic medical records system to attempt to get this available at the point of care, to give prompts so that one doesn't necessarily need to know that in the last few months there's been an approval for an exon 14 rearrangement. And also to work to make certain that these panels are covered by the third-party payers. Both ASCO as well as some of the other professional societies are working with the payers to be able to have these panels paid for in the appropriate clinical settings, and then also to work with them so that you can have the targeted therapies available for the appropriate indications.
So, Bruce, I know that this advance fills you with hope and has not only helped to define your career, but has allowed you to help define an advancing standard of care for the disease that you focus on. But even with that said, what part of this keeps you up at night? What is the thing that makes you most worried about either the delivery or the advancement of this area of work?
So the part that concerns me the most is being able to disseminate the information in a meaningful way to the people at the point of care who are taking care of these patients. One of the things in our field is that we've seen a gradual evolution from having general oncologists to being more specialized, and we did it decades ago in the academic centers. We've seen the movement from community-based practices commonly to hospital-based practices, and seen the size of the practices grow. One of the things that we hope is that as it grows, people become familiar with the diseases that they treat, but to know what's meaningful. And when I say meaningful, to know that a specific genomic change can be effectively targeted. For instance, in my own practice, we've seen the length of time a given drug works, and the most dramatic example is with the ALK rearrangement I talked before, where the initial treatment of it worked for 10 months, and now it looks like it's going to work for somewhere around 3 years or even longer in some of the patients. And that really transforms the care for that group, and we would like that information to be disseminated rapidly to the point of care for the people that commonly see and treat these patients. It's one thing to be able to manage the common diseases where the genomic changes get a large degree of publicity, but there's other things of, when it becomes to unusual things like Merkel cell tumors where some checkpoint inhibitors look like they may work in a subset of these, and having that information disseminated so people either know that there is a treatment where there wasn't one previously available, number one, or number two, know where to send the people to be able to get those treatments that have been developed for these unusual tumors.
One of the points that is I think assumed in our discussion today, and I don't want to leave it without just mentioning this, is that although we don't have time today to focus on this, it would be a whole other podcast. The patient experience is obviously very different with these therapies. In most cases, not all, they represent either lesser toxicities or at least different toxicities. And I think that the excitement that the audience should hear in our discussion relates to that, as well as to the better targeting of the cancers and the better outcomes that we hope to see. So with that, I do want to thank you, Dr. Johnson, for joining me today for this ASCO in Action podcast.
Yeah, it's been a pleasure. It's nice to go over this and try to give an introduction to our theme of the annual meeting, and hopefully this will become more widely available both to our practicing oncologists as well as to our patients.
So if you're inspired to watch the full-length webcast of ASCO's recent Precision Medicine event, and I encourage you to do so, you'll see, again, an in-depth discussion with an esteemed panel, but very importantly, you also see the experience of a patient given voice on stage. Then you can go and visit asco.org/state-cancer-care, and you'll find there a link to our webcast. It's worth your time, I think. So until next time, I want to thank you for listening to the ASCO in Action podcast.